Good morning children. Welcome to your chemistry online class. Last class we have discussed about glucose and fructose. So in today's class we are starting a new biomolecule, that is amino acid and proteins.
And amino acids. In our human body, the 20% it consists of proteins. Proteins are the large complex molecules that are critical for normal functioning of our cells. They are essential for the structure, function and regulation of the bodies, tissues and organs. And proteins are made up of smaller units called amino acids. So, amino acids are the building blocks of proteins. They are attached to one another by peptide bonds. And using this peptide bond of amino acids, it forms long chain of proteins. And we'll see what are amino acid and how it is classified. And amino acids are very interesting compound which having both amino group and carboxylic group. So we know amino group is a basic group, carboxylic acid is an acidic group. So it can act as both acid and base. And we can call it as alpha amino acids. If the amino group is attached to the alpha carbon, then we can call it as alpha amino acids. Here you can see the picture of alpha amino acids. So every amino acid is having a alpha carbon and which is bonded to carboxylic group and amino group, that is NH2 group. And it is also attached to hydrogen and aryl compound. So it is a chiral compound. Then we'll see the classification of amino acids. So we can classify it as hydrophobic amino acids and hydrophilic amino acids. If it is dissolved in water, then we can call it as hydrophilic. If it is not dissolved, hydrophobic. And mainly there are 20 amino acids based on their nature of their R group. So based on the polarity, we can classify it as hydrophobic and hydrophilic. And hydrophobic further classified as non-polar. They are non-polar in nature. And if the R group attached to, we have seen that amino acids having 4 different types of groups, and there is an R group. If the R group is an alkyl, we can classify it as we have the examples glycine, alanine, valine, leucine, isoleucine, methionine, proline. In your textbook, a detailed column of these amino acids are given. And if this R is aromatic, then it is phenylalanine or tryptophan. And if it is hydrophilic, then it is polar. This is polar. And polar can further classify into neutral, acidic and basic. How we can classify it as neutral and acidic and basic? It can classify as neutral and basic and acidic. If we check these amino acids, if they have only one NH2 group and one COH group, it is neutral. Because acidic also there, basic also there, so it will be neutral. For example, tyrosine, serine, cysteine, etc. Acidic. If we take aspartic acid, in aspartic acid, there are 2 COOH group, that is acidic group, and 1 amino group. So it have, most probably it have the acidic nature. So such compounds are known as acidic. Such amino acids are known as acidic amino acid. For example, glutamic acid and aspartic acid. Then what is basic? If number of basic group, that is amino group, is more than acidic group, then it will come under basic. For example, lysine, histidine and arginine. And the next type of classification is based on essentiality. We know amino acids are very essential to human body. Not only human body, every organism needs amino acids. So there are two types of amino acids that are essential and non-essential. Essential amino acids are the amino acids which you need through your diet. We have to take it in the part of our food because our body cannot make them by itself. So we have to take it through diet. Whereas non-essential amino acids, these are which will produced by our body. So no need to take it through diet because it can synthesized by our body. So for example, essential amino acids are histidine, isoleucine, leucine, etc. Non-essential alanine, asparagine, etc. So these are the main classification of amino acid based on the number of hydroxyl functional group and based on the essentiality. Then we discussed that amino acids are combined through peptide bond to give proteins. So what is this peptide bond? That is, we know in amino acid there are two type of functional group, that is amino group and carboxylic group. So if an amino group of one amino acid and carboxylic group of another amino acid bonded together, what will happen? A water molecule eliminate from that, and it will give an amide compound, that is a peptide bond. So in the time of protein synthesis, the carboxyl group of amino acid at the end of the peptide bond, amino acid, for example, here you can see that the carboxylic group of one amino acid and amino group of another amino acid combined together gives a peptide bond, and it will eliminate one molecule of water. Here you can see the peptide bond. Such bonds are known as peptide bonds. And we'll see structure of amino acids. Structure of proteins. So the sequence of proteins is written by the DNA of the gene that encodes the proteins. That is, there are DNA and RNA and all that we'll see in next class. If there is in DNA also there are amino acid groups. If there is any change in the sequence of amino acid of DNA, there will be change in the protein. Even if there is one change in one amino acid, the protein sequence will affect the overall protein structure and function. So it is very essential the order of amino acids. For example, if instead of alanine, if there is glycine, entire function and entire structure of protein will change. So to understand how a protein gets its final shape or conformation, we need to understand the 4 levels of protein structure. There are 4 types of protein structure: primary, secondary, tertiary and quaternary. So the primary structure. Here it is a simplest level. It is there we can see a sequence of amino acids. In a polypeptide chain, it's a chain-like structure. A sequence of amino acids are there. And sometimes you can see sulfur-sulfur bond between that. SS chain. So, the primary structure of proteins refers to the sequence in which amino acids are arranged in proteins. Each protein has a specific sequence of amino acid units which is critical its biological activity. So, it is the simplest level. Here, amino acids are arranged in a sequence manner. For example, glycine, lysine, valine, glucogen. Here you can see a order. If there is any different in this order, for instead of glycine, if there is cysteine, the entire function of this amino acid will change. This is the example. If you take, for example, insulin, A and B chain is there. It is an example for a primary structure. Then secondary structure. It is a folded structure. There is a attachment of polypeptide due to interaction between atoms. It is like a pleated sheet. There are the secondary structure further classified as alpha helix and beta pleated sheets. Both structures are held in shape by hydrogen bonds. For example, here you can see beta pleated structure and alpha helix structure. Alpha helix like a helix-like structure, like this, a helix-like structure. Beta pleated is a sheet-like structure. Here it is due to the hydrogen bonding. The beta pleated structure. In this type, the peptide chains lie side by side in zigzag manner. The peptide chains are like arranged in a zigzag manner. Here you can see that. And it give a flat sheet type structure. For example, keratin having beta pleated structure. This keratin is present in our hair and silk of silkworm, etc. It is having beta pleated like structure. Then alpha helix structure. In this structure, the polypeptide chain are coiled up to form a spiral structure, and it is stabilized by intermolecular hydrogen bond. Intermolecular hydrogen bond between C double bond of one amino acid to the amino group of another amino acid, that is NH and C double bond O bond. There is O and H bond, that is intermolecular hydrogen bond is there. For example, myosin. Myosin in muscles is an example of alpha helix structure. And we'll move to the tertiary structure. It is a three-dimensional structure of proteins. It is primarily due to the interaction between R groups of the amino acid, and the interaction is hydrophobic interactions in this. That is, amino acid with non-polar hydrophobic R groups cluster together in the inside the protein, leaving hydrophilic amino acid on the outside. So inside hydrophobic amino acids, outside hydrophilic amino acids will cluster together to give a three-dimensional structure. And also disulfide bonds also you can see here. That is tertiary structure. Here you can see sulfur bonds and the outside hydrophilic amino acids and inside hydrophobic amino acids. Then quaternary structure. When multiple polypeptide chain subunits come together, then proteins attains quaternary structure. If there are multiple polypeptide chain, they are like a cluster-like structure. Here you can maybe alpha helix and beta pleated all together will come gives quaternary structure. Example for quaternary structure is hemoglobin. It is having two alpha helix structure and beta pleated structure. And there is another important property of protein is denaturation. On heating or treating, what will happen is with chemical reagent, it will change its shape. For example, protein having its own unique shape, and if the temperature or pH and if it is exposed to chemical, the protein's environment is changed. These interactions may disrupt the structure of the three-dimensional structure of protein and turn back to unstructured string of amino acids. If a protein having linked structure, if it is reacted with heat, pH or etc., it will change to amino acid strings. So it loses its higher order structure. The protein is having a very good higher order structure, but it is not changed as primary model. It is denaturated. It is became not functional. It is having a function, but after heating or a pH change or chemical reaction, it will form like this structure, and it is not primary structure. It is called denatured, and they are usually non-functional. It is having no functions. For example, maybe due to biological activities. So this phenomenon in which proteins undergo changes in physical and biological properties without affecting its chemical composition under the effect of heat or chemical reagent is called denaturation. For example, the coagulation of egg white on boiling is an example of denaturation. So during denaturation, secondary and tertiary structure are destroyed, but primary structure remains unaffected. Only secondary and tertiary will be affected by denaturation. Example, egg white, egg coagulation. So what are the functions of proteins? Proteins are source of energy required for various metabolic processes. Some proteins are enzymes, act as biological catalyst. Hemoglobin is a protein, act as a oxygen carrier. Insulin is a protein, and so there are so much properties of protein. It is very important to our daily life. Thank you.